Saurabh? Hello,
Yes, ma'am.
Saurabh.
Yes, ma'am.
Okay, starting. Very good afternoon, everybody. Myself, Dr. Rimi Dotto, on behalf of THK Jain College family, would like to cordially welcome all of the participants, our respected principal madam, coordinator and members of IQAC, our honorable speakers and my colleagues on the last day of this 4-day online lecture series entitled Virtual Journey through Botanical World, organized by the Department of Botany in association with IQAC of our college. We are very thankful to all the participants for their active participation and a lot of attention that would surely help us to enhance the excellence of this session and make it more successful. But we feel very pity for a very few participants who are paying more interest for filling up the feedback link to get certificate than listening patiently to this valuable, very informative speech of such eminent speakers. Therefore, a very humble request to them from not for asking for feedback link repeatedly, and please help the organizing committee to pick up the valuable questions of participants who are listening with attention. We will surely provide the feedback link at the end of the session. We would again like to apologize for any technical glitches that may arise due to internet connection during the session, which is beyond our control. I would again like to heartily thank all of you for joining us on today's session. Now, I would like to request Dr. Saurabh Ghosh, my colleague and member of organizing committee, to continue the session.
Thank you, ma'am. Am I audible?
Yes, yes.
Okay, so dear participants, before we start the session today, we have some announcements to make. Please listen very carefully. Since today is the last day, I request all of you to pay attention to the following announcement, as we will not be able to repeat it. We will be giving the link for feedback form on the entire program today during the end of the session after the interactive session. Please do not keep asking for the form. It will be surely given on time. It is our sole responsibility to provide you with the feedback link, as these feedback link will be used for generation of your e-certificates. Therefore, it is very important for you to fill up with the correct spelling of your name with the correct salutation, as it will be reflected the same way in your e-certificate. Also, you should correctly enter the name of your institution with proper spelling or abbreviations, capital or small letters, etc. Whatever you enter as your name or the name of your institution will be taken up by the software and published in your e-certificate directly. No correction request for publishing of certificates or correction of certificates will be entertained by the organizing committee. The form will remain active for 24 hours from when the link is shared. So there is no hurry to fill up the form. Hence, please fill it up correctly. As there are about 1,200 participants, the issuing of certificates will take some time. It is a software-oriented program that issues the certificates and that issues 100 certificates randomly per day. So it will take at least 2 weeks to issue all the certificates, if not more. We will be highly obliged if you wait patiently for the certificate till 2 weeks. After 30th August, if any of you do not receive your certificate, even after correctly submitting the feedback form, I repeat, if you do not receive your certificate even after 30th August, in spite of correctly filling up and submitting the feedback form, you may mail with your issue to the organizing committee in the provided mail ID in the brochure. No mail regarding e-certificate will be entertained before 30th of August. Thank you all participants. So welcome to the fourth and final day of the online lecture series entitled Virtual Journey Through Botanical World. I must mention the enthusiasm with which the participants are actively participating every day has been driving us through. Yesterday, we had an overview on the physiological process of plants and their genetic control. The lecture by Dr. Ashim Kumar Ghosh was highly informative and very interesting. Such detailed slides with so elegantly constructed animations and describing each aspect of the topic so vividly in such a short time period was highly appreciable. Thanks a ton, sir. Moving on, today we are going to have a session on the most important applications of plant biology, tissue culture and biotechnology. The title of today's session says, From Totipotency to Transgenic Plants. Totipotency is a property that has opened wide dimensions in plant science research and development. It is the exploitation of this very feature of plants that the whole subdiscipline of research, tissue culture, is in existence. Tissue culture is a science, and again, it is a form of art that requires a very high amount of skill and precision and demands perfection and attention in every minute detail. It has multiple applications in the field of agriculture, crop improvement, horticulture, pharmacy, and no doubt it plays the most dominating role in biotechnology for the service of mankind. Genetic modification in plants or raising transgenic plants, popularized as GM crops, is another important research field to overcome the never-ending demand for the ideal plant crops. Ideal as in with great qualities. Scientists have been working since the last few decades to achieve the most out of nature, to achieve ideal. An ideal crop denotes the best characteristics such as good productivity, high adaptability, pest resistance, and more, tolerance to salt, drought, and n number of things. The zeal of researchers to achieve this has opened wide windows to transgenic research. Vivid knowledge of plants, biochemistry, genetics, molecular biology, biotechnological techniques, and above all, dedication and love to plants is needed to achieve this ideal. So today, to guide us in our journey from totipotency to transgenic plants, we have with us Professor Gaurav Gangopadhyay, a dedicated scientist in the field of molecular biology and biotechnology. So let's hear from him directly about how amazing the working with plants is. I request the convener of the program and head of the Department of Botany, Dr. Rimi Datta, to introduce the speaker and welcome him. Over to you, ma'am.
Thank you, Saurabh. It is really a great pleasure for me to introduce Gaurav Gangopadhyay, Professor in the Division of Plant Biology, Bose Institute, Kolkata, India. He is a plant scientist having a feel for the minute details of the plants and the belief that understanding the molecular basis of subtle variations can enhance a plant's performance in the field against biotic and abiotic stress. He has been in the field of plant science research for the last 30 years. His area of research interest is marker assisted plant breeding, plant biotechnology, and molecular biology. After graduating from Presidency College, Kolkata, and post-graduation in botany from University of Calcutta, he did his doctoral work at Bose Institute. He has a postdoctoral research experience for over 12 years. Dr. Gangopadhyay has 73 research publications, 67 NCBI Jain Bank submissions, and 12 submissions in high throughput sequencing database sRNA, that is Sequence Read Archive. His H index is 15 and the RG score is 24.27. He has supervised 5 PhD theses, 7 MSc dissertation theses, and presently 4 doctoral students are working with him. He is a reviewer for the scientific journals like the Journal of Agricultural Science, Cambridge, In Vitro Cellular and Developmental Biology, Plant Genetics Resources and Crop Evolution, and PLOS ONE, to name a few. Saurabh, over to you.
Yes, ma'am. So I cordially welcome Professor Gaurav Gangopadhyay to begin with his lecture. A gentle reminder to the participants, please do not unmute yourself or present anything during the course of the lecture as it adversely affects the session. However, you are allowed to put up questions for the speaker to be discussed during the interactive session in the chat box of both Google Meet and YouTube live platforms. Sir, if you are ready.
Yes, I'm a success. And all other staff members of Department of Botany and Jain College, I am thankful to all of you for giving me this privilege. And thank you, Dr. Datta, for your nice introduction, though I do not know whether I'm worthy of it. Anyway, so already Saurabh has told you about the title and the main issue of this today's lecture. It is from totipotency to transgenic plants. So just to begin with a different note, so I'm showing you the pictorial view of the main campus of Bose Institute. The day before yesterday, you have visited virtually Acharya J.C. Bose Botanical Garden at Shibpur, Howrah. So this is another place, in fact, the abode of Indian science, the temple of modern science, the heritage building founded by Sir J.C. Bose almost 103 years back. And this building, in fact, it is the first building. It is in the north direction. That's why we call it as the North Building. And I do not know whether the principal ma'am is present today. And this is this lab where she used to work. I used to work in the first floor. So those were the days, but still flowers are blooming and everything is all right. We are maintaining the heritage and Bose Institute is going from places to places. So this is another view. This is the another view where you are seeing the Samadhi, where the ashes of Sir J.C. Bose is preserved. And in every occasion, we start with paying our homage to this place. So I am also paying my homage to our founder, Acharya J.C. Bose, on this webinar. This is another view. This is the south building of the main campus. And this picture, while I took, to create a depth of field, I just placed 2 plants here, and probably you can recall, some of you, that these large leaves, basically, these are the leaves of teak plants. Just remember this thing. It will be recalled in this lecture. So now another view. It's a panoramic view, which I took the photograph few days back, and the west building here and the north building here. And now you can probably see, I do not know the presentation of the screen sharing, that clouds are there on the western side. And that's why this picture is so contrasting. And these clouds were blooming, and these clouds were increasing. And on 20th May, something happened which turned our life topsy-turvy, and it was the super cyclone Amphan, which devastated Kolkata, Bengal, and many parts of India. And Bose Institute was also devastated, mainly our main campus, our Muthamgram campus, Darjeeling campus, Falta campus. Almost all campuses were devastated during this Amphan. And the following day, and in fact, this particular picture was taken on that night after that storm was stopped. And this photograph was taken by a security person who is staying inside. The lawn was flooded and everything was destroyed. So it was a very painful sight for the loyal people like me who were here in Bose Institute for so many years. And after 2-3 days, when I ventured to go to Institute, almost it was a voyage through a forest. That Calcutta was a peculiar scenario. And on the main gate, that huge trees were fallen. So we had to start afresh. Apart from this pandemic, COVID, that was another issue that Amphan and all the staff members, the contractual staffs, they worked hand to hand overnight. And after a few days, one thing I noticed that few days back, all those logs were cut, their pieces, they were kept aside. And this picture I took just 7 or 10 days back when I was in the lab. I suddenly saw that fallen teak plants, in fact, those teak plants were almost at the age of my research age. In my earlier days, I used to work on tissue culture of teak plants. In Bengali, it is called Shegun. Scientific name is Tectona grandis. You know, it is a prized timber tree. And in that time, during that time, and before that, we used to transplant it a few in our campus. Mostly, we gave those saplings to the forest departments. So one of our children, this is almost 30 years old, he is falling, but I suddenly saw that new shoots are coming out, piercing that woody timber. So possibly it indicated a ray of hope of life. From another angle, I took another picture. Almost all the cut logs, all the fallen pieces, they are again sprouting. Probably it is giving the ray of hope, the undaunting spirit of the plants. Suddenly, I remembered one sculpture. This particular sculpture is present. I am showing you that sculpture. This sculpture was present in before Plant Tissue Culture Lab of University Agricultural Science, GKVK, Krishi Vishwavidyalaya Kendra, Bengaluru. It is a masterpiece from an artist, Mr. Jagannath Jakarpalli. It signifies the capacity of survivability of a living organism, and probably it is best expressed in plants. A tree failed is shown to have an undying spirit of life, hence new shoots have sprung up from cells of senescing stub of a tree. So how symbolic this sculpture was, and in fact, this thing we see here and there, but probably this feeling was kept in the artist's mind. So he put this remarkable sculpture. And what Saurabh was telling, the concept of totipotency, that the potential of giving rise to a whole plant from a single somatic plant cell, it stemmed from the vision and wisdom which emanated from the experience of Dr. Gottlieb Haberland's work, a German scientist. In 1902, he first coined the term totipotency. For his pioneering works paving the way for tissue culture research, he is rightfully recognized as the father of plant tissue culture. So let us pay homage to this artist of this beautiful sculpture, and let us pay our homage to the father of plant tissue culture, Haberland, before going straight into the technical issues. Okay, this was the introduction. Probably you liked it. Now, coming straightway to the technical issues, as I have already told you, that totipotency is the ability of a plant cell to regenerate the whole plant. And it is the basis of regeneration. It is the basis of de-differentiation. Only a plant cell is equipped with this enormous potentiality, which the animal or even the bacterial cell do not have. So these pathways of regeneration, basically it is a twofold, that regeneration can occur either through embryogenesis or through organogenesis mode. I will discuss in detail about each of these things, and just try to remember that both this embryogenesis and organogenesis can occur either through direct or indirect mode. All these things are a bit of technical tissue culture thing, but I will try to clarify those points. And nowadays, these things are being taught even in undergraduate botany classes. So each and everybody now almost in this field are acquainted about this, terminologies. But let us revise this thing. Now, I am showing you a very important information. This information was taken from the famous scientific journal. Probably you know the two most ancient scientific journal, one from Europe, it is Nature. Now there are many journals of Nature group, and another is from America, United States of America, it is Science. And Science, on its 125th volume, they took a worldwide opinion poll from the scientists of diverse field. It was like, sending nomination about what the issue was, that what we do not know till now. Though we the scientists boast a lot, we can do this, we can do that. But today in this scenario of COVID pandemic, probably it is very clear that still we are at the foot of the nature, and even the highest technology is struggling to cope up with the deadly disease. But still, science and technology is a process. It is a religion to the scientist, and it is a growing process. Not that today's science is absolute. It is always it's a continuum. It is a vibrant thing. But the most wonderful thing which I noticed, because among all the unanswered questions, very few questions was raised from the plant's perspective. The reason is very simple. We take plants as taken as granted. Even the plant scientists are second grade citizen to rocket scientist or physicist, or this is so complex, but this is true. Even our students who they do not get very good postdoc abroad, but even almost all our plant science students, they switch their field to cancer biology. But often we forget that we depend on plant, and almost we know from our childhood that we are dependent on plant for our food, for our existence, for our respiration, that we take plants taken as granted. But only one poor question, but most relevant question came that how does a single somatic cell become a whole plant? What does it mean? It means it is fascinating to fathom the concept that a somatic cell, not the product of the union of two gametes. We know a seed germinates. We know a gamete, two gametes fuse to form a zygote. Everything we know. But does a single somatic cell taken from a leaf, taken from a stem, taken from a root, can it give rise to a whole plant? Yes. That's the beauty of plant cell, and that's the intricacy of totipotency, or total potential, the potential of a single cell to give rise to a whole plant. So this is still now an enigma. How does a single somatic cell become a whole plant? And this was categorized in that issue among the most unresolved mystery till now. So now, let me start with one, say, geometric figure. Why I am showing this thing? Because often I have to take few classes in our postgraduate courses for plant developmental biology. And when I start, I always start with this slide. I have taken this slide from my class lecture. It is the image of a sphere. So if I ask you how many symmetry can be generated from a sphere, the answer will be infinite. A symmetry, as a sphere, remains same on rotation or reflection in any axis, passing through its midpoint, and therefore it shows infinite planes of rotational and reflection symmetry. Now you may be thinking that why I am showing this sphere. Yes, if you think twice, it is the mother of all structure. All the embryos, be it plant or animal, initially it's a sphere. It starts from a sphere. And a sphere is so plastic that it can give rise to any structure. And the embryo of a plant or an animal, it always a sphere initially. That's why, say it God, say it Mother Nature, say it evolution, he or she has created sphere as our starting point. Now, coming to embryogenesis, how embryo is developed in plants. In this slide, there are two pictures. One is written zygotic embryogenesis and another is somatic embryogenesis. Zygotic embryogenesis, it is the usual thing which we know, that a zygote gradually gives rise to a cotyledon, a seedling, and later on which gives rise to a whole plant. And if you see, it is a picture from a standard plant biology book, Theis and Ziger, that it starts with a, it starts with a octant stage. If you see here, this small circle, because initially it was single cell, that after cell division, from 1 it becomes 2, gradually it becomes 8. So from 8 it is octant stage. It gradually divides in all planes. So it becomes globular. Now the beauty started. It started an invagination from the midpoint on the upper portion, and it gives rise to a heart shape. And this heart shape, the two edges, are essentially the future two cotyledons. And if you see the torpedo stage, now the structure is more or less destined to give rise to a seedling, because these upper two will give rise to cotyledon, and this red portion, it will give rise to future shoot apical meristem. And here, again, the lower portion, the blue colored, it will give rise to the root end. And now if you see the comparative developmental stages of somatic embryogenesis, so what does it mean? This embryogenesis is the resultant of the cells of somatic cells which have not undergone gametic fusion. And here again you will see that it starts from globular stage, which is a sphere. Though in this picture it is a bit ellipsoid, but at the initial stage it is a sphere, and then globular converts into heart and consequently give rise to torpedo and cotyledonary stage. Well, these are theory, and these things are found in each and every plant developmental biology textbook. Now let me share you some of my experiences with this somatic embryogenesis, and those experiences are fascinating because, say, these pictures, essentially these pictures are scanning electron micrograph derived from scanning electron microscopy. The various structures which you are seeing here, basically these are all different stages of somatic embryo. I first personally encountered these structures during, say, late 1997, 98 to 2000, 2001. In one of my earlier papers, when I was a postdoc in 2002, I published these structures. Basically, it is the embryogenic callus of Chenopodium. It is not such important about the material. You just consider the phenomena. And if you see here, if you see this structure, this is, since touchpad creates problem, I should have used the mouse. Anyway, so I do not know this structure. If you see here, this is the resemblance of a heart, complete heart, which we see in our human on any vertebrate heart. And all those structures, you can see here, there are different stages of development of heart, torpedo, and cotyledonary stage. Now, these structures were observed in a population of embryogenic callus. Now, for the students, I am saying, because there are many college teachers and senior persons, what I have found from the participant list, but for the undergraduate and postgraduate students, I am saying this thing, that this is an example of indirect somatic embryogenesis. Now, if you remember that I have told you that both somatic embryogenesis and organogenesis can occur either through direct or indirect mode. So why I am calling it indirect? The reason is very simple. If you see the flow diagram in the left, you will see that I have written leaf, and then I have put an arrow, and then I have written callus. So what does it mean? That from leaf we have induced callus. Callus is an assemblage of unorganized cells. Now, these unorganized cells, due to proper media manipulation and all other physical and chemical nutrient, it has turned embryogenic. So all the cells of this callus are now ready for giving rise to embryos. So these embryos are coming out from somatic cell of a leaf. So that's why they are called embryogenic callus. So this is not occurring directly from leaf. So there is an bypass or intermediate stage of callus. So that's why we are calling it indirect somatic embryogenesis. And if you can nurture all these somatic embryos properly in proper tissue culture condition and media, all of this somatic embryo will regenerate to a plantlet. So what Saurabh was telling about this totipotency and the implication of it, its implication is huge. So each and every somatic cell can give rise to a potential somatic embryo, which can give rise to a potent plant of desirable trait. So here lies the beauty. So it was the first experience of mine with somatic embryogenesis as early as 2002. Now I am showing you some pictures of one of my papers in the same year when I standardized that a single cell, what I was telling you, because in that previous slide, what I have shown you, basically it was a mass of cells of a callus. But for many experiments, we have to start with a single cell. And from where you will get the single cell, definitely you have to start from a plant part. Say, for example, it is leaf. You have to convert it into callus. Now, this callus, normally it is grown in solid media or semi-solid media, because this media is gelled with agar. You know it. Now, if you do not give agar in the media and just make a little bit of improvisation, you can grow these friable cells in liquid media also. If you don't put agar, so basically the media is water-based, so it will be a liquid media. And for making it fragile, you have to put it on a shaker so that all the cells get proper nourishment and respiration. And then we call it suspension cells in suspended condition. And in this cell suspension, all these cells initially, if you see the image A, it is a total sphere. So it is the globular structure. And in C or D, you can see that heart and torpedo stages are coming. So it is fascinating. When I first witnessed this thing under phase contrast microscope, it was just amazing to see how a single plant cell is converting itself to a potent embryo. And again, you have to put back those cells to solid media to prepare to develop microcallus, and from that callus again regeneration takes place. So again, so for the students mainly, I am showing this thing. So it is the example of cell suspension mediated indirect somatic embryogenesis. I think it is clear. So this type of webinar, actually, it is not very much interactive. So I cannot see your faces. I cannot share you. But probably you are getting my point. So let me move to the next slide. So now from this black and white era, let me come to the age of colors. And this is from one of my recent works in 2014 and 2018, where while you do not think that I was just obsessed with this somatic embryogenesis and doing all this thing in different materials. Not actually the case. Actually, this is in relation to different research programs, different research projects, where one stage or other, we have to use this thing. And in every case, we have to put the document for our publication so that we can convince the reviewers or the readers or the future generation that it was done in the right way. So here you can see the image A is an non-regenerating callus, pale yellowish. And after proper media manipulation in B, you can see here that profuse plantlets are coming out in this regenerating callus. And when this callus was put under scanning electron microscope, you can see particularly in D that somatic embryo in heart stages are formed. So these are real beautiful images. And thanks to this advanced technique of electron microscopy. Again, another example, though this is not yet published. So this image I have taken from one of my students' PhD thesis. So it is an example of somatic embryogenesis in pineapple. presently I have a project on pineapple transformation, and in connection to that work, if you see here, that here you can see, sorry, I must not use that touchpad. And if you see the A in this photograph A, you can see from a large 1 or 2 pineapple leaves, many tiny micro shoots of pineapple are coming out. So that is the beauty of tissue culture. So it is showing repetitive organogenesis from this inner sheathing leaf base or meristem of the leaf. And we can detach each and every micro plantlet. This was a tiny plantlet, tiny pineapple plants. And from this tiny plantlet, we have again produced callus. Those callus again turned embryogenic. Okay, till now I have told you about all the indirect modes. So each and every cases here, this embryogenesis has taken place through callusing. So now let me show you some evidences of direct embryogenesis. I must agree, this is not from my work. Right now, I do not have a picture of my own, of my own work for direct embryogenesis, though presently I'm working on a project on tea, camellia, and on that material also direct embryogenesis takes place, but I'm not showing that pictures for obvious reason. This is an example of direct somatic embryogenesis I have taken from one leading paper. And if you see in this photograph A, it is immature zygotic embryo. And from this zygotic embryo itself, a huge number of globular somatic embryos are coming through repetitive embryony. So there is no intermediate callus stage, and from one single zygotic somatic embryos are coming out, and if can be handled properly, all these somatic embryos are capable of giving rise to plantlet. Now, what is the implication of this? And take it from me, this direct somatic embryogenesis mostly takes place in case of woody plants. I am showing you an example. I have also taken this image from internet. It is not from my own work. So probably you know, in temperate countries, in Canada and in many parts of America, forest fire, seasonal forest fire is a serious issue, because due to many reasons, the conifer forest, due to presence of resin, and of course due to anthropogenic activities, forest fire takes place, which causes huge forest loss. So the foresters of those advanced countries, always they rely on advanced technology of replenishment of those forests. And essentially, they rely on this direct mode of somatic embryogenesis. And if you see the schematic image here, I am not touching the touchpad because it is giving rise to the next slide. So here you can see in the right side that ovule containing immature ovule, immature embryo. And from that immature embryo, zygotic embryo, a number of somatic embryos are coming, and they have made this thing totally automated. They put this regenerating zygotic embryo, which are giving rise to somatic embryo, like this embryos, they have put it in bioreactor. And bioreactor, but these plant bioreactors are very intricate because you have to maintain each and everything. So in these bioreactors, after certain duration, these globular stages, they all synchronizingly transform into heart stage, then they again go to torpedo stage, and finally the germination takes place. So it's a fascinating way of showing the application, large-scale application of totipotency and somatic embryo. So by one shot, you can produce the plantlets of desirable plantlets, and after hardening in greenhouse, you can transplant to the forest. So the foresters of the Western countries, they use this thing regularly. Now I'm sharing my experience with some encapsulation or artificial seed, which I worked almost 15 years back, and that time this work was quite popular, and even now it is one of my highest cited paper. It was published in Current Science in 2005. You can see here that in this left image, that tiny microshoots of pineapple are emerging out of small bead-like structures. And the students probably recognize this thing from their textbook or their knowledge, that these are called, we call it synthetic seed or artificial seed. So we can even store each and every tiny plantlet for quite a long time through different processes in different temperatures, definitely lower temperatures. Even we can cryopreserve it to even minus 196 degrees Celsius. But in my case, it was just low temperature. And after proper treatment, all those beads of sodium alginate, I will not get proper time to discuss that encapsulation technique. Probably you know all this thing, but this technique, in this technique, in place of agar, we use alginic acid and sodium alginate. In fact, that salt is used. And these beads, when these beads are formed, when sodium is replaced by calcium due to cationic exchange, and these beads are basically representative of calcium alginate beads. And in fact, in these beads, there are nourishment for plants' hibernation for longer period. And when you want, you can produce the plantlet. So you can use it as well. So this technique, which I used, it became quite popular, I must say, with modesty. And later on, I got certain option from this work that I am going to share with you. Now, if you remember the title of my talk, From Totipotency to Transgenic. So till now, I have shown you the example and potential of totipotency, but now I am trying to convey the message how totipotency is related to transgenic plant production. And here I have put cuttings from my two papers, soft copies of my two papers. One was published in 2009 in Plant Cell Tissue and Organ Culture, a leading tissue culture journal, where we were first able to make genetic transformation, what we call nowadays recombinant DNA, not this simple genetic transformation of pineapple, Agrobacterium mediated transformation, using a novel encapsulation-based antibiotic selection technique. Now onward, this thing will be a little bit more technical. I will try to convey. I hope I will be able to reach to all of you. But this technique became so popular and time tested that after say 5 or 6 years, I was invited from Springer publication to put this thing in proper way to make a standard protocol so that anybody who has a little bit idea of plant cell and tissue culture can 100% with 100% surety can make pineapple transgenic. So this is a series which is called Springer protocols and Agrobacterium protocols. The researcher and the teachers, they All of you know probably this thing. And here we tabulated all the nitty-gritty things, how to make this pineapple transgenic using encapsulation technique. Just I'm showing you some glimpse of it so that you will get an essence about it. Say this leftmost picture is an image of which I have taken from Wikipedia. So just a representative figure. So we started with the top portion, which we normally cut while we consume pineapple. And if you see in detail in the LS or the cut view, you will find that there is a central triangle-like white space, which is actually the shoot apical meristem. And we started our culture with that meristematic zone, and from there, and if you see the step by step, we were able to produce innumerable pineapple microshoots. And from there, we, in fact, some of these pictures I have shown you in my earlier slide, and this is a more detailed view. And from those microshoots, after media manipulation, those were converted into lumps of embryogenic calli. So now, why we are doing all this thing? Only for the academic sense? Not really, but for the application side, because the persons who have a little bit idea about this genetic transformation, if we can expose these somatic embryos or the small callus species to the Agrobacterium culture, where the desired gene is there within the plasmid. The desired gene may be for, say, any disease resistance or herbicide resistance, whatever may be what Saurabh was telling. And during this stage, this Agrobacterium will simply infect that plant cells. For here it is the pineapple cells, and each cells have the potential to give rise to embryo. So why we are doing this, or what will be the outcome? Because when this Agrobacterium will enter this plant cell, pineapple cell, they will make homologous recombination with the genome of pineapple. And it is a chance process where our desired gene, which is placed in the plasmid, artificially we have created that plasmid. We call it construct, gene cassette, many things. This is technical molecular biology term. And that gene from that cassette will be placed here and there in the pineapple genome. Now the issue is that how to find that particular cell or embryo or potential portion of callus where that transformation has taken place. And in fact, till now, this is a big challenge, because you can almost routinely make Agrobacterium mediated transformation, but you cannot routinely obtain a large number of putative transgenic plants, because sometimes there are no transgenic, sometimes there are false positive. So what we did, we actually worked in this area, that we, after infection, we put those cultures in small beads, calcium alginate beads, and we put the selection antibiotic, hygromycin, kanamycin, everything within this bead. Why? Because in this small spherical milieu means environment, there will be a stringent selection, because that gene is co-localized by antibiotic selection gene, with the assumption that the transformed cell will survive even in the place of high concentration of antibiotic, which a normal plant cell will not be able. But in normal case, what we see in culture practice, many cells which are not properly exposed to this selection condition, they simply escape. And often plants are coming out from that portion, and we think that it is a transgenic plant, but in the long run, it proves to be a non-transgenic one. So it makes the efficacy of the system very poor. But using our technique, we we increased the efficacy of transgenic of up to 20%. Probably you must be laughing that 20%, is it a real percent? Take it from me, the success story of plant transgenic is just 2%. People are happy if they get 2 good transgenic plant from 100 recombination event. So it was an achievement. So that's why they gave me the honor to put it in the proper way. And the side left panel, the green, bluish portions, basically these are called gas positive. So probably time is not right to discuss all this technical issue. Anyway, but from totipotency to transgenic, you can probably have an idea how this somatic embryo can give rise to transgenic plant. Now let me come to the other aspect of regeneration, which we simply called organogenesis. And this is an old picture from our work, which was published again in Plant Cell Tissue and Organ Culture as early as 1997. In fact, again, I am saying it, not we were doing everything for the sake of getting somatic embryogenesis or organogenesis. It was every time we were doing this thing for a specific issue, specific objective. And in that time, we were working. In fact, it is a portion of a work of my wife, Dr. Shungita Gangopadhyay. I was the second author of that paper. Anyway, but this paper, in this case, we were trying to make salt-tolerant rice through somaclonal variation. That time, transgenic was not very popular. It was quite a new technique in 1995 or 6 or 7. That time, people used to try somaclonal variation, protoplast fusion, anther culture for crop development. So it was a very success story. We make salt-tolerant rice through somaclonal variation. But the reviewer or the other people, they were not believing our story. So we have to give the real images of the somatic, of the callus-mediated regeneration. And here, why I am calling it organogenesis, because here there was no somatic embryo. In fact, what happened that the callus piece, if you see here from the white callus, gradually it is coming green zones where the shoot buds came. And it is the case of organogenesis. And there was an intermediate callus phase. So that's why it was a callus-mediated organogenesis in rice. And again, in recent year, it is a work in connection with another program where you can see from a single leaf of tobacco. Tobacco is a model material of plant tissue culture. You can see from the base petiolar portion, so many plantlets are coming. So these organogenic techniques have huge impact on transgenic production. Now I am coming to 2 of the recent works of Dr. Saurabh Ghosh, our very beloved student who is the faculty member of Jain College presently. Though Saurabh is not directly my PhD student, but he is more than that because he was our product. I mean, he did his post-graduation in Bose Institute, and we were all his teacher. And Professor Shomit Shikdar was his PhD supervisor. But a significant portion of his work, Saurabh's work, was stuck because though he got some excellent information, some excellent finding, that he had found a protein, RIHSPRO2. It stands from HSPRO2, say it is a name of a protein, heat shock protein. RI stands from Roripa indica. It is the name of Nasturtium indicum. And the Professor Shikdar's group is long working on controlling aphid in mustard. You know, aphid is a big issue in mustard. But there is no aphid-tolerant genotype in mustard or rapeseed mustard. But there is a wild Roripa where no aphid infection takes place. And in fact, Saurabh, through his elegant biotechnological and molecular biological work, he isolated and he identified that resistant protein which is present in Roripa. I am putting it in a very simpler way, which is a very promising candidate to control mustard aphid, because Roripa belongs to same family of Brassica. It is Brassicaceae, or earlier it was crucifery. Now, though it was done by Saurabh, and he published it in a very good journal, Arthropod Plant Interaction, in 2018. But he had to show it. He had to show it making a transgenic in mustard itself, so that people can believe that protein, if can be overexpressed in mustard, which is sensitive to aphid, that if he can overexpress that protein in mustard, then mustard will be resistant against aphid. So you can understand the challenge of science, the demand of science. Then we cannot boast anything without showing evidence. So at that point of time, Saurabh again came to my lab, and we worked long on the regeneration of mustard or brassica, because there is always a problem of regeneration. And thankfully, we were successful, and it was a very intricate paper. Almost everything was done by Saurabh. Just I helped him to induce, though this image is small, if you see the image B and C, you can see here that from one tiny cotyledon of mustard, innumerable plantlets are coming. So it is an example of organogenesis. So what we did, we actually took cotyledons of mustard and co-cultivated in Agrobacterium suspension. And in this Agrobacterium, there was plasmid, and that plasmid contained that RIHSPR02, that gene, nematode-resistant gene. I think it is clear to you. And that plasmid, that cassette, was successfully integrated into the genome of the mustard. And since it was a very efficient organogenic system, from a single cotyledon, multiple shoots are coming out through organogenesis. After, say, gas study, or say PCR or technique, or say southern hybridization, Saurabh got very good percentage of putative transgenic, which showed the presence of RIHSPR2, and in the long run, he proved that this transgenic mustard, which he had developed, that is resistant to aphid to some extent. It is quite a good discovery. So I was proud to be a part of that work. So in fact, that is again an example of how the concept of totipotency goes up to the transgenic level. Now I have come almost to the end portion of my talk. I will not give you a big lecture, long lecture. It is tiring both for me and for the audience. Now, again, I am coming back to that issue which was addressed in that journal Science, that it is a mystery. How does a single somatic cell become a whole plant? It is still an enigma. And how people work on it, and why people work on it. Because probably you recall that while they were introducing me, they were telling something very important, that tissue culture, it requires a very good eye, a very good intuition, and often it is, people say it is a bit of empirical. It is not so quantitative like other science. But what I can tell you, that it requires a sense, a feel. But science the hard science actually doesn't depend on feel. If you can do one thing, the other people also will be able to do the same thing, then that science is a real science. If you can do one thing, you can make a discovery, but other people cannot, so your science is not reproducible. So if you cannot decipher the mystery of totipotency, if you cannot boast that I will make a system which will give 100% regeneration, any plant, any transgenic will give, say, 30 to 40% positive transgenic, then it will be a success story, and then it will be its commercial application. And why till now the plant scientists cannot do this, are not able to do this? Because it is still an enigma. They do not know the mystery, the interaction of the molecular factors behind this in vitro morphogenesis. What the orchestra is being going on inside, that is still not known. It is not the play of a single gene. So that's the main issue. So how the people approach this thing? So I'm giving you some idea and some of my recent works on this aspect. So this is a paper which I downloaded just yesterday while preparing the slide. It is just a review, not my review. It was published in 2020, this year itself. Basically, it is a review on genes, proteins, and other networks regulating somatic embryogenesis in plants. That I was telling you. Basically, it is a networking of innumerable molecular factors, genes. And if you see the green ellipse inside here, you will find that there are innumerable genes written here, the abbreviation capital form. And I have made one red circle. It is showing WUS. I will go into detail of this WUS. And if you see here somatic embryogenesis has been shown, can either through direct mode or through indirect mode. Now you can understand all this thing. And be it direct, be it indirect, it will go through the same stages: globular, heart, torpedo, cotyledonary stage. Now the issue here is why we cannot make predictable transformation or predictable transition from globular to heart or heart to torpedo to 100% of the cells. Why there is no synchronization? So that's the research field. So people all over the world in this field of research, they're trying to accumulate information about the genes, the transcription factors which correlate this thing. And once this whole story is known, and in the right you will see another red marked gene is there, ACRK1. So I will tell a little bit about these two genes in two of my recent research programs before I finally conclude. And this is another work which I took from another review yesterday. So it is another approach where people make proteomic approaches through total proteome analysis. People want to find the elusive proteins which are behind the somatic embryogenesis. Now, this is other people's work. Now, let me come to my work. So, before going to this, I am just showing you one slide, which probably you remember, you have seen it from your school days, that the onset of shoot morphogenesis, basically it is the initiation of shoot apical meristem. And this picture is known to you from your school days. And nowadays, we do not call the shoot apical meristem, we call it SAM, shoot apical meristem, but this leaf primordia, often we designate this leaf primordia as plastocron, what I have written in the center, because it is very difficult to delineate the first leaf or the second leaf, because meristem development is a very dynamic event. So nowadays, what should I say? People call this thing as the time interval between successive lip primordia plastocron. And the rightmost picture, you can see that it is colored in different places. So till now, people have deciphered which particular genes or factors are mainly playing in which zone of shoot apical meristem, or in first plastocron or second plastocron, like this. So people work in this way nowadays. And just 2 years back, while I was working on this WUS, or its full form is USHEL or USCHEL, it is a transcription factor which regulates the shoot apical meristem development. And in this scanning electron microscope image, you can see one circle is there, where I am showing that tiny shoot apical meristems are coming from this lip base region. And this image was a tissue culturist's delight. In fact, it was the best stage I have ever captured. And you can see here, the inside, it is the shoot apical meristem proper. This is, say, lip primordia 4. So let me show you. This is 4, this is 3, this is 2, this is 1. So you can understand the phyllotaxy, how these leaves are coming out, lip primordia are coming out after successive time intervals or plastocron. And we correlated these events with RNA and reverse transcription quantitative PCR analysis. We call it expression analysis. I am not going into very much detail. This QRT-PCR nowadays is very popular due to COVID, for COVID detection. We also do this thing in our plant system. And I was ultimately, rather not I, we were ultimately able to prove that how USCHEL transcription factor is controlling that shoot apical meristem or that plastocron development. And this is a schematic, this is a model where we have shown that how this how this USCHEL protein is interacting with DNA, because it's a transcription factor. Similarly, I worked on another transcription factor, SERK. If you remember that another red box, what I have shown you, SERK, it is the full form of somatic embryogenesis related receptor kinase. And I identified those that kinase from two-dimensional gel electrophoresis of different developmental stages of pineapple, and through Multi-Toff analysis. This to explain this thing, I need another lecture. I do not want to do so. Anyway, so these are all high-throughput proteomics technique, and through molecular modeling, we were able to prove that how somatic embryogenesis receptor kinase is controlling somatic embryogenesis. So this is just some example how people decipher this thing. And finally, just 2 or 3 slides more. Very recently, within this lockdown, one of my papers was published. I was working on an orchid. It is called vanilla. If you can see here that there is different stages of vanilla regeneration, and somatic embryos, from these somatic embryos, plantlets are coming. And from different developmental stages, we did isozymic analysis. It is a traditional technique. And also we did two-dimensional gel electrophoresis. This is not one-dimensional gel electrophoresis, because one-dimensional protein gel has certain limitation. It can identify peptide, and that peptide can be derivative of different proteins. But if you run that gel in second dimension, the first dimension is isoelectric focusing, and second dimension is STS page, then those proteins will be visualized as individual spots. And these individual spots, when those were exclusive of, say, globular stage, of, say, heart stage or torpedo stage, then you can probably say that this particular protein is responsible for converting globular to heart, or in a very broad sense, from a non-regenerating callus to regenerating callus. And if this type of information pouring on continuously, so within few years, people will be able to make predictable regeneration and transgenic production. So this is almost all. This is my last slide. My acknowledgements to my students and colleagues of Bose Institute. And this is an opened fruit of tea, about which I was telling you that from this broken nut-like fruit of tea and this white embryo, now I am working on somatic embryogenesis of tea in vitro tissue culture plants in connection to another program. So maybe after few years, if everything goes all right, I will keep that lecture going to your college. So that's all I have to say. Thank you so much for your patient hearing.
Thank you, sir. And you are actually most welcome to our college. If conditions are, such pandemic conditions are over, and both you and we will be safe if you visit, then we will surely call you up. In fact, we always do plan to have sessions in our college, but this pandemic situation has really, they have embraced our hands. We can't help. Anyway, sir, I must say that thank you, obviously, but it was such a well-organized lecture, which graded gradually from simpler to complex things. But again, those complex phenomena appeared so simple with your wonderful slides. The presentation was really fascinating, and the personal experiences.
Shaurabh, don't flatter me. Shaurabh, don't flatter me.
No, I'm not flattering, but actually those personal experiences that you shared along with the introduction of the lecture with the Bose Institute campus, it was great.
It was Dr. Mousumi's campus, and I have shown Mousumi's lab there.
Yes, I will acknowledge you. I will definitely acknowledge you. I'm really happy to listen from you after a long time, Gaurabda. Really.
Oh, nice.
Okay.
Great.
Okay,
let Shaurabh speak.
I'm coming to you. I'm coming to you all after. Okay.
Let Shaurabh.
Surely. And with your permission, sir, I would like to open the session for interaction, as there are many questions from our participants.
Yes, of course. So okay. What I have to do, I have to listen and answer, or I have to see something?
No, you have to just listen. I'll request my colleagues and members of the organizing committee, Dr. Orpita Mukherjee and Dr. Anindita Singh Roy, to start with the interactive session.
Please.
So, Orpita, over to you.
Thank you, Shaurabh. I'm audible? Am I audible?
Yes.
Thank you, sir. Thank you so much for such a beautiful, vivid, and informative lecture. Especially the beautiful virtual journey through the campus of Bose Institute. We, along with all our participants, were delighted to see it. And especially, sir, the undaunted spirit of the. So that was really very nice to see. As usual,
okay, come to point.
Yes, sir. Please, we are flooded with number of questions. I would like to take as many as I can. I'm taking up some of them. First question is, what are the factors that makes plant cells totipotent?
Just a minute.
Sir, am I clear?
Now it is clear. Yeah, please.
Okay, sir. So the question is, what makes plant cells totipotent and animal cells not?
There is a breakage of sound.
Can you hear me?
I'm here. I'm hearing you. Please put it again.
Yes, sir. Yes, sir. What are the factors?
What are the factors?
Okay, ma'am. Is it clear now, ma'am? Is it clear now, sir?
Yes. You continue.
What are the factors that makes plant cell totipotent and animal cells not?
Oh, great question. Asked by definitely some knowledgeable person. And in fact, I think
Yes,
he
sir.
knows
It's
the
one of our
answer.
speakers, sir.
Huh? He is the first speaker.
So I think it's one of our speakers, sir.
Oh, actually, it is my examination,
Yes,
actually. He or she knows
no,
the answer. Anyway, I'm just joking. So the essential difference of a plant cell and animal cell is that plant cells are bipolar. So like the Mother Earth has North Pole and South Pole. Plant cell, though those cells are spherical, but there are two hidden poles inside. And this attainment of polarity from a spherical structure, that is another story. About this, I often take class in my developmental biology, how a spherical structure attains polarity. So why I am saying about polarity? Because this two poles will be the future shoot and future root. Now, people are again working on it for a long time, and you must understand that the animal cell doesn't have any polarity, apolar structure. And the plant cells, their polarity is so important, and people are working on it because after a few years, if we survive this COVID pandemic, maybe we will be at Mars or Moon, where again we have to depend on food on plants, and we have to grow plant there in vitro because there will be no agriculture in that condition. And in that condition, the polarity of the cells will be disrupted because the roots are positively gravitropic. So one pole, they sense gravity. But there will probably going to another topic, but the answer is that question is that why plant cells are totipotent and animal cells are not? Because plant cells have the unique bipolar structure, which if grown properly with proper nourishment and physical condition, they can give rise to two end. One end give rise to shoot and other root, which the animal cells cannot do. Over.
Thank you so much, sir. So there's another question. Why transgenic plants are not yet introduced in most of our crops, oil, vegetable or spice yielding plants in India?
In fact, no transgenic plant has got that license, and this question is rather a socio-economic political question, not in the purview of a scientist. But I can tell you about the technical things, because the most popular or most talked about transgenic which came to our place was BT cotton or BT brinjal. So you know all this about this Bacillus thuringiensis. But the socio-economic issue, the environmental issue, everything was at stake. And you know, being so advanced, Europe are still against GMO or genetically modified organisms. In America, United States, they use transgenic crops, and in the products of transgenics, while in our case, say, we nowadays in our food packet, we see green spot or red spot, like veg or non-veg, they mark it as, say, GMO crop, non-GMO crop, organic crop, like this. So there are many issues, but if it is done properly, if the science is good, if the technology is good, and if the field trial is rigorous, it is done in confinement, of course, transgenic plant will be coming in future. We have to accept it. Over.
Thank you, sir. Thank you so much. So another question is, if we use particle bombardment for delivery of genes in an X plant, is there any chance of hampering any other gene functions like flowering or fruiting? If so, how can we recognize and fix it?
Yes. again, these are very technical questions and people are taking my exam almost. These are very tough questions they're asking me. Okay, I'm trying to answer it. Okay, there is no conflict between electroporation or particle gun or agrobacteria-mediated transformation. Just a minute.
Yes, sir, sure.
Hello? Hello?
Yes, sir. So you're audible.
Sorry. So there is no difference between agrobacterium-mediated transformation, electroporation, particle gun. All are techniques to deliver foreign DNA into plant. And particle gun, electroporation, they deliver naked DNA, while in case of agrobacterium, the vector is a bacteria. So since agrobacterium and plant has long association, natural association, their probability a little bit more, but in some cases agrobacterium doesn't work properly and people has to rely on particle gun and electroporation. Now the second part of the question was that if this particle gun induced gene delivery can hamper other expression of other genes. Yes, that is true. That is that often happens. Often happens. Gene silencing often happens. Multiple copies integration. So that's why the obtainment of a large number of successful, transgenic plant, the true transgenic is a challenge. So it is the big challenge. Often there is gene suppression, many things happens. So after say T2 or T3 generation, we nowadays in earlier days if we could have make one transgenic, we were happy. Now in fact development of a transgenic doesn't assure you a publication. Now you have to start from there. You have to first decipher the molecular biology of your transgenic plant, whether it has the right cassette inside, whether it has brought anything else. This is, we call, from the genetics point of view, we call it as linkage drag. Because theoretically, transgenic, in case of transgenic, we introduce a single gene. In case of breeding, while this is due to linkage phenomena, a number of genes come inside, which are often eliminated by backcrossing. But even in case of transgenic, very closely placed gene can come together due to linkage drag. So first you have to prove that the only that gene which you have targeted have inserted successfully and it has not hampered the activity of other genes. So it is a challenge. It was a nice question. Over.
Thank you so much, sir. So there's another question. Can we use intercalary meristem for bamboo as explants for indirect somatic embryogenesis? This is the question. I'm clubbing it with another question which is very similar, that what is the future prospect of bamboo transgenics? And we can develop more bamboo plants. There might be a reduced conflict between man and elephants, since bamboo being the main source of elephants. I don't know how much this relates to that, but I'm placing this question before you.
Again, it's a very good and technical question from knowledgeable people who are working on this aspect, because these are normal questions. These are not very normal questions. These are very specific questions, and with the person who has asked this, he knows the issue. Anyway, I am giving you the answer. You can start with any meristem, because in case of bamboo, often if you have to make a clone of a proven bamboo cultivar or variety, maybe you cannot get the terminal meristem. So maybe you can use that intercalary meristem. But the chance will be, in fact, bamboo tissue culture is a very tough job. And in 1990, early 1990, Professor Natgora from NCL Pune, National Chemical Laboratory Pune, he first shown that bamboo can be grown in vitro. And in fact, he was able to show in vitro flowering of bamboo. In fact, that paper came in Nature. Normally in plant science, we simple tissue culture, it doesn't come in nature. But that time it was a breakthrough paper. Bamboo tissue culture is possible in our institute also. Our ex-senior professor, Professor Amita Pal, and his group used to work on it. I have seen bamboo tissue culture, but I can tell you one thing. Growing bamboo through tissue culture and replenishment of bamboo plantation through it, I do not know how far it will be cost effective. It can be practical for any conservation of a rare bamboo, but for a general bamboo, you have to rely on, say, vegetative propagation techniques of bamboo plantation. That's my answer. Over.
Thank you, sir. So some of our students are very interested to know what is the basis of encapsulation and why mostly sodium alginate is used for this purpose. I think some of our students are interested to know the encapsulation process.
Yes. Encapsulation means encasing a somatic embryo or microshoots for certain duration within a closed environment, a milieu. And that's why you have to put all the essential nutrients. Like, say, it often we call it quote-unquote synthetic or artificial seed, because in seed we have the embryo in its dormant state, or say, vibrant state, but that embryo is nourished by the endosperm. So you have to feed the somatic embryo through different means. And in our case, it will be that salts, vitamins, hormones, which we normally use in tissue culture. Now, if we make a solution of, that liquid media, everything, you have put everything in that liquid media, but you are not allowed to give calcium chloride, which is otherwise given to all tissue culture media. But while you are doing encapsulation, initial media will be devoid of calcium chloride. So you are putting one by one everything of, say, MS Murashige scoop media. So you have made a liquid MS media, and then you have a suspension of your somatic embryo. Now you just mix 1:1, and then in that suspension, you have to add, in place of agar, the molten powder of sodium alginate or alginic acid, which is obtained from seaweed. Now, this is suspended cells or somatic embryos are in an environment which contains sodium alginate and all other nutrients of MS media minus calcium chloride. Now you have to take this suspension in one hand, say your left hand, and you have to take calcium chloride solution outside in another hand. I cannot show you. If I can have a board, I can exclusively show you. Now you have to, it requires a great skill. And you have to, now it definitely has automation, but in our case, we do it manually. Even till now, I can do it very effectively. You have to mix this thing very quickly, and as soon as you mix it, what will happen, that sodium will be replaced by calcium. It is called cationic exchange. So calcium will drive away sodium and calcium alginate compound will form. And sodium alginate was a semi-solid viscous thing, but calcium alginate will be a crystal white bead-like beautiful structures. And here, all those things will be entrapped. So, and then you have to make certain manipulation here and there. And then this technique goes on. So I think I have given the answer. Over.
Yes, sir. So you have explained so beautifully. Sir, there are many questions more, but I think I'll just take one more question and then I'll pass on to Anindita. So is there any future prospect to produce UVB stress-tolerant transgenic plant?
Okay. My question to the participant: why you want to do this? Because there is a UV stress. Okay, I understand.
She's working on it. I'm not sure.
Okay. Just let me think. I haven't thought about it. So now I can give you the answer, the essence of it, though it will be a bit blah blah, not very specific. Essentially, you have to work on the phytochrome system of the plant, because all this photosynthesis, you have heard the illuminating lecture of Dr. Ghosh yesterday. So photosynthesis is dependent on light, and UV is another spectra. That's all right. But how that, so what should I say, the waves, the electromagnetic waves are captured by the antenna molecules of the photosynthetic receptor, that's a big mystery. So nowadays, the light reaction pathway is more or less deciphered. Many things can be manipulated. So I do not know whether there is any specific gene which, say, can perceive ultraviolet B, and probably tinkering with that portion of the genome may rise to. Nothing is impossible, but first we have to understand the science very beautifully. Then comes technology. Often we jump to technology. I'm not saying BTech is bad, but BSc is better. Please just you take it from me. The answer is like that.
Yes, sir. Yes, sir. Thank you, sir. Thank you so much. I would now request my colleague, Dr. Anindita Singh-Roy, to please take up some questions from the YouTube live stream. Over to you, Anindita.
Again, question. My God.
Yes.
Okay, go on.
Thank you, Urpita. Am I audible?
Yes, audible.
Thank you, sir. Thank you very much for such a wonderful presentation. And so just few questions from YouTube. So first question, is there any opportunity to obtain secondary metabolites by tissue culture?
Oh, of course. That portion I haven't touched, because then it will be again the case of yesterday. So not that. That is another huge prospect. Secondary metabolites are the very happening thing. And plant tissue culture, especially the suspension cultures, are used. And there are many success stories in case of rosamarine, agmalicin, vincristine. But again, there is in, say, early or late 90s, it was a very prospective story. But people somehow have stopped this research a little bit, because the bioreactor, because these things go on through bioreactor, and maintaining the plant bioreactors are very tough. And very few companies worldwide, some Japanese companies are producing sikimic acid, rosamarine, as I have already told you, through this secondary metabolite. But nowadays, people often go for genetic manipulation, or often go for elicited treatment. And because secondary metabolite is a big issue of plant, because say one particular metabolite is accumulated in root itself. So for just extracting that thing, you have to propagate a large number of that plant. It requires labor, say land, everything. But if you can have a handful of very prospective culture of that root cells, and if you can put it in bioreactor, definitely you can get. But till now, its commercial applicability is not at par with microbial bioreactor, I must say. That's a good question. Over.
Okay, sir. Moving on to next question. Which type of cells be used for somatogenesis?
I meant somatic embryogenesis. Okay, so because it is a very, say, honest and question. I mean, very theoretically, I can say any cell, but that is not the case. In fact, while we teach, we say something, but while we practice, we know what is to be done. Theoretically, all cells are totipotent, all plant cell can give rise to somatic embryo. But say, if somebody was asking if I can use intercalary meristem of bamboo, yes, theoretically it's possible. But I know the pain and anguish of surface sterilization and getting that juvenile tissue. So my answer is that it is better to take the mostly meristematic tissue where it is present. Say if you can start with cotyledonary explant, it is excellent. Sometimes hypocotyle gives a very good result. And if you take, say, terminal meristem, if you can take axillary meristem, don't go, always go for juvenile tissue, not go for, say, older tissue. So more juvenile tissue, more chance of regeneration. That's the answer.
Okay, sir. Moving on to next question. There are a few questions related media preparation for somatic embryogenesis or for plants requiring different types of media. So one question from amongst I'm asking, sir, how to formulate media for different plants?
Very good question. Because till now there is no universal media, though often media is becoming synonymous with MS. Everybody says MS. But if you go back to your library, nowadays this is all digital library, to see the pain of Professor Murasige and Skoog to formulate that media. It was published in Physiologia Plantarum in 1962. Prior to it, there was Gambox media, White's media. But it was a painstaking work. So what they have formulated the bare minimum, the essential requirements, the macro, micronutrients. But there is no rule which is applicable to all plants. You have to make trial and error for all the genotypes. And that is the problem and beauty of tissue culture. That's why it is still called an enigma. But those materials which have more commercial prospects, which are mostly used by industrial purposes, they in that case, those media are more or less patented. And even nowadays, if you go to any standard catalog of high media or Sigma, you will find that they are selling media, orchid media. They are saying, say, anthurium media, banana media. So those media have become 100% standardized. And suppose you are working on a plant which is growing in the virgin area of, say, Ladakh. So definitely you have to work on it. But mostly you have to work on it, growth regulators and the minor addenda. The major things are already established. That's my answer.
Okay, sir. Another question. Do we have transgenic pineapple plants fully formed by this process in India?
No, I am working on it. In fact, one question was there regarding the prospect of transgenic plant, which I have already answered. Now, the big issue is that whether you are incorporating any foreign gene in it. And from there, the battle begins. The BT gene comes from Bacillus thuringiensis. You know the story. I am not going to repeat it. Now, in one of my works, I was working on rice, and one of my students have, and she is listening today. I have seen her name. Hi, Sharonda. She is in England, and Shananda worked on phytic acid biosynthesis in rice, and she introgressed gene, phytase gene from Escherichia coli. Now, if you want to introduce this rice in India, there will be an uproar. Oh, she has introduced one bacterial gene, and maybe it will be deadly. But people, oh, Shananda is saying, hello, sir. Okay, hi, Shananda. So now I'm not going into her work. Her thesis will be over within few months. Now, if you want to overexpress something in your own system, then it is again a transgenic. Say, what I am going to do, I am trying to overexpress a gene of pineapple. What does it mean? The gene is there itself. And we have found the function of that gene very clearly. So now if we can overexpress those genes, so the end product will be more. So that is my target. And we have seen that this gene have a potentiality to to tolerate one certain fungal strain. So by this way, I'm working on to develop a transgenic pineapple, which will be able to tolerate soil-borne pathogen. That's my answer.
Thank you, sir. I think this was the last question. Not last question, there are many questions, sir, but I apologize to the participants due to time constraint. However, sir, it was really kind of you to answer, and thank you once again for such wonderful presentation. Now I would ask Shorab. Shorab, are you there? Over to you.
Yes, I'm here. Thank you, Anindita. And thank you, sir, for being so patient with the questions. And there are still a number of questions, but really we cannot take all the questions.
Shurab, you know I am always patient. Shurab, you know I am always patient.
Yes, but sir, we are getting impatient with the large number of questions. Even there are requests for you, sir, that arrange a whole day webinar with Gaurav, sir. So I don't know.
I'll be there.
Let's see if we can manage
you finish because your participant will be angry now.
Thank you, sir. So to continue with the session, we have been receiving many feedbacks and comments, valuable comments in the chat box, and also we have received many mails and WhatsApp chats. So today, since it is the last day, so we would like to have an online session for the feedback from some of you who can say for us. We have amongst us many delegates and faculty members of distinguished institutions. So would I request, I would like to request Dr. Shoma Haldar ma'am, if you are with us, please can you say something about your experiences about the sessions? Ma'am, are you there? Hello. Hello, Shoma Halda, ma'am. Okay, I think she is not with us for now.
No.
Oh,
She is with us. She's muted. Ma'am, please unmute yourself. Ma'am. Shoma Haldar, ma'am, please unmute yourself. Okay, Shurab.
Yeah, shall I continue, ma'am? So I would request another faculty or senior faculty of Jogomaya Devi College, Sir Sushovan Vera.
Hello.
Yes, sir.
Hi, Sushovan.
Come on.
Thank you. Oh, you go on.
Thank you. Thank you, Dr. Bose, Saurabh Bose, for giving me the opportunity to say a few words about my experience. At the outset, I would like to thank you all, including your principal and organizing committee of the TKH Jain College, for organizing such a big web lecture series with a large number of participants from all over India as well as abroad. In that sense, it is really a great success. Actual reasons for my participation in this lecture series was simply to refresh myself during this pandemic situation, and another reason was to hear recent updates from at least two of the speakers whom I know from my college days. Number one, Dr. Asim Kumar Ghosh, my teacher, and secondly, Professor Gaurav Gangobadhyay, one of my best friends till college days. So anyhow, the first lecture on arrival of the first greens by Dr. Sen Sarkar was really a beautiful uh, with excellent photographs and modernized timescale. And it was really a very good experience for me to know about different algal genera of past. Really good experience for me also. Uh, Dr. Singh, on the day two, Dr. Singh presented our botanical garden from a different perspective. We all came to know about double coconut, its pollination and post-pollination development in our garden, and newly introduced aroma garden, which was totally unknown to me, and also newly developed app for virtual journey through botanical garden, uh, which will be very good attempt by the authority as at VSI. We have also seen the post-Amphan situation of botanical garden, that is uprooting of several valuable plants as well as replanting, etc. Yesterday's lecture on plant physiology by Dr. Ghosh was really an experience for me, at least, as well as uh, many of us. Sir, I enjoyed your lecture, very nice lecture with nice animated presentation with explanation of every aspects of plant life within just 1 hour 45 minutes. That is in a very nutshell, which was really a challenge for you also to cover all the aspects of plant life within such a packet form, what you used to call in Puria form. Thank you, sir. Today's lecture by my friend Dr. And Professor Gangopadhyay is really very nice as well as informative. I know him from my college days, and I also know that he is a very good orator, dedicated scientist for a pretty long time. His nice presentation on Bose Institute at the outset, and then the totipotency to many intricate aspect of plant tissue culture, and finally transgenesis enlightened us in a very luminous way. Thank you, Professor Gangopadhyay, for your fascinating lecture, at least for all, not myself, your friend.
Thank you, Sushavan. Thank you.
Lastly, I would like to request the college authority to host similar type of webinar series for benefits of student as well as faculties in future. So with this word, I thank you all. Good evening and happy Independence Day. Thank you all. Thank you, Gaurav.
Thank you, sir.
Thank you.
So your words
Sir,
were
for your
really
suggestion.
inspirational, and we will actually like to again plan for such events. Yes, ma'am, you were saying something. Sorry, ma'am. Hello? Hello? Ma'am, you were saying something? Okay. So, is Dr. Soma Haldar, ma'am, are you there?
Ma'am to ache. Yes, yes.
Am I audible?
Ma'am, please. Yes.
Audible.
Yes, ma'am. Yes, ma'am. Please continue.
Firstly, I must thank you all the conveners of this webinar to give me the chance to tell few words about the experience of this 4-day-long program of your college. The most important part, what I feel, of this webinar series is that this was totally directed to enlighten different fields of botany, which is very much important today, as most of our students, they cannot realize the importance or the future prospect of this subject. When they read the subject, they enter in their UG course. So this webinar series is really nicely presented by all of the speakers. Firstly, I must mention the Dr. Nida Jain Sarkar, who was from Kolani University, from where I have also done my UG and PhD studies, enlightened us with a new area of research field about the fossil algae, which was totally unknown to me also. And I feel that we all know that the algae are the most important main source of fuel, and that area of research will throw some light there. Next day, I enjoyed really very much about the lecture by Dr. Basant Singh of BSI, who has represented really a nice virtual journey through our favorite botanic garden, which will attract all the students for the subject botany, knowing the diversity of the plant kingdom. I have shared that slide to my third semester students, as this year we don't know whether we will be able to visit their garden in proper time. So it will be enjoyable to them. At least they will see it, they will look all the plants, and they will come to know about the speciality of that garden. Next, our respected sir, Dr. Ashim Ghosh of City College, gave insight into the plant physiology and how all the metabolic processes are controlled genetically, which opens a new area of research to the youngster's mind. And lastly, today's scientist, Dr. Gaurav Gangopadhyay from Bose Institute, where I also did my thesis work, enlightened the students the application part of the subject, which will encourage all the students to know more about the subject. Personally, I really enjoyed all the lectures and already shared many of them to my students and colleagues, which I feel they will also enjoy. Congratulations to all the conveners once again for arranging this webinar series and expecting many of more exciting topics like this in near future. Thank you once again.
Thank you, ma'am. Thank you for the inspirational words. While organizing, we were very much concerned about how the students will feel like. So we have been receiving their feedbacks as well. I call upon Srijan Mukhopadhyay. He is a student of Botany Honours from Serampur College. Srijan, are you with us?
Yes, sir. Am I audible?
Yes, you are. Please share your feedback on the session.
Yes, sir. Good evening to everyone. I personally would like to thank everyone, the organizing committee and our dear and respected speakers for conducting such a beautiful webinar that consists of 4 days. On the first day, Dr. Jain Sarkar took us to a journey where we dug deep into the Earth's past and we came up strata by revealing fossils. And we also came to know how the first greens that colonized not just the sea, but then they moved on to the land, and then how they diversified. And the tiny glimpse of their diversity was expressed on the second day by Dr. Singh when we took a virtual trip to the Botanic Garden. And it was mind-boggling. I wish I was there present physically, but certainly this virtual initiative was really brilliant. Then on the third day, when we have seen how life has been diversifying, we entered deep into their cells and tissues to figure out how is this diversification exactly working. Through the plant physiology webinar by Mr. Ghosh, and it was beautiful and beautifully arranged how hormones regulate their growth and development. And then today we see on the webinar by Dr. Gangopadhyay how beautifully we can manipulate some things, let us say biological, if I may use the word weapons or tools, to actually manipulate and bend biological processes to our benefit. That is harnessing the power of nature. And this 4-day journey was very beautifully presented to show us how life has evolved and how we are evolving along with life. And it fills me with gratitude to see such dedicated and beautifully conducted presentations. And there were so many participants who were constantly questioning, and the questions were answered very beautifully. Overall, it was a very beautiful experience. Thank you so much for this experience. I would love to have such experiences in the future as well. Thank you.
Thank you, Srijan. So let's hear it from another student, Shucheta Mazumdar, if you are there.
Yes, sir, I'm there.
Yeah, please continue.
Hello.
Please continue
Hello,
with
good evening.
feedback.
Hello, good evening, everyone. I'm a student of THK Jain College from Department of Botany. I would like to say a few words. When I first heard about this virtual Journey to Botanical World webinar, I was very excited. On the first day of the webinar, as the session began, our HOD ma'am and the professors gave thorough introduction of the webinar and its content, which made me more enthusiastic to know what was going to happen. When Dr. Neera Shen-Shorkar ma'am started presenting the slides on the imaginary views of Earth, that very moment grabbed my attention. And slowly she continued. When she was describing about the time period, my interest grew more as I had no idea about the eons, eras and epochs. The pace of the lecture with which she carried on was understandable by a student like me, as I could absorb every new fact that I came across. On second day session, Dr. Basant Kumar Singh sir took us to a virtual journey to the botanical garden, and that was an amazing experience. When sir was explaining about the curious plants present in the garden, especially the great banyan tree, I was feeling if I could be there and see the canopy through my eyes. And that very moment came the bird's eye view of the canopy, as if sir felt it and put it up on the slide. Also, the restoration projects that have been going on after the destruction caused by Amphan was really eye-catching. And finally, when Sir told us that they are introducing an app for virtual visit to the botanical garden, it was a great initiative. I'm looking forward for the successful launching of the app. The third day session was presented by Dr. Ashin Kumar Ghosh Sir. The slides were very good and informative, especially the cycle. But there was something about the genetic control of physiological process which was a bit difficult for me to understand. Yet the slides made it much more lucid. When I would study physiology in future, these slides would help me academically. And today, at the last day session, when Dr. Gaurav Gangabadhar sir started the lecture with showing the view of Bose Institute along with the sculptures, that was very fascinating. The way Sir shared his experiences and explained everything with so much detail, it's much more interesting. The slides were very unique and informative. Overall, today's lecture was mesmerizing and a great experience. Well, I have said a lot, but please bear with me for a few more seconds. I would like to thank the organizing committee for arranging such a great and informative session for us, and along with the interactive sessions at each day of the webinar. I would like to organize the committee and show my gratitude towards them for giving us such a great opportunity to learn during this pandemic. That's all. Thank you, sir.
Thank you, Suchita. Okay, so I think that will be all, though we are having many more students, but again, time is a constraint. So I would now request our respected principal, Madam Dr. Moshumi Singh Sengupta, to please say a few words.
So today is the last day of 4-day online lecture series. So I'm really surprised, and at the same time, I'm really happy to see that you have organized this seminar. I mean, this online lecture series so efficiently. I am really grateful to all the faculty members of Department of Botany, Dr. Rimi Dotto, the head, Dr. Shaurabh Bose, Dr. Anindita Singh Roy, Dr. Urpita Mukherjee Banerjee, and other teachers also. Really, you have put a lot of efforts to organize, for a successful completion of this seminar. And thank you. And during this journey of 4 days, actually, we have been enough fortunate to listen from eminent speakers who are the really expert in this particular field. I am thankful to Dr. Neera Sen Sharkar, Dr. Ashim Kumar Ghosh from City College, and Dr. Ratan Singh from Indian Botanic Garden, and Dr. Professor Dr. Gaurav Gangopadhyay from Bose Institute. So really, it is a great learning experience, and I am sure that all the participants, all the audience have benefited out of this. The lecture. So, and I'm thankful to Dr. Shushopon Bera, sir, for your valuable feedback, sir. Really, and of course, Shoma madam, we are really will definitely we have a plan to organize such webinar series for the students as well as faculty members, research scholars. Now, on a personal note, I'm very much thankful to Dr. Professor Gaurav Gangopadhyay because he's from Bose Institute, and I am also a part of parcel of that prestigious institute where I have pursued my PhD and on plant molecular biology and biotechnology. And he has such an informative lecture session today, and he's a very good teacher. He's not only a very good scientist. I have seen him in very close close association, that he was not a very dedicated scientist, really a great teacher that we feel today. Because at that time when I started my research career at Bose Institute, we are really fortunate to have a senior like Professor Gangopadhyay. At that time we don't have any Google information, we don't have any the online scope of going through the research articles and everything, and we have to depend totally on our seniors. And really, I'm very much thankful to Gaurav and I'm really happy to hear from you after a long time. And overall, this 4-day webinar series has seen success, and it's of course due to the hard work not only put by the teachers, faculty members, and support from the technical support from other various departments, computer science department, and also the audience, the participants. Really, thank you audience, thank you participants for your cooperation. And we have definitely we are also planning to organize such web series for all academic fraternity who are engaged in the botanical research and associated with this subject. And one thing just I want to share with you, the speakers that you have grown interest, even the students what I followed from the chat box, that you have the speaker said you have been able to grow interest of botany for students like Department of Commerce. I have seen one of my the few students are there who have commerce background. They are also listening to this beautiful lectures. So that is definitely, we are really happy to share with you all this. So with all this, I express my immense thankfulness to the governing body members, to our secretary sir, Sri Lalit Kumar Kankaria, president sir, for their active support to organize this type of various programs, from using the online platform series by various departments of our college. The history of this college is not very old, and it's started its journey in the year 2006. And from that, just we are trying progress in various fields. Okay, so with your I'm very much thankful to all of you, and thank you for your patience. Thank you.
Thank you, ma'am. We will really, with your support, again plan such programs for the college and from the Department of Botany. Thank you, ma'am. So I now request the convener of the program, Dr. Rimi Datta, head of the Department of Botany, THK Jain College, to propose the vote of thanks for the 4-day online lecture series, Virtual Journey Through Botanical World. Over to you, ma'am.
Thank you, Saurabh. Now we have finally reached the end of the online lecture series entitled Virtual Journey Through Botanical World. I deem it to be a great honor to propose the vote of thanks to all who have helped us in making these 4 days online lecture series such a resounding success. First of all, I would like to thank all the eminent speakers for gracing the whole session with their tireless effort and by sharing their precious knowledge with all of us. Dr. Neera Sen Sarkar had introduced us with the evolution of early greens on Earth on the first day, while on the second day, Dr. Basant Kumar Singh took all of us to a wonderful virtual journey to AGC Bose Indian Botanic Garden, Kolkata. On day 3, Dr. Ashim Kumar Ghosh introduced us with the various aspects of plant physiology with his great expertise. And today, Dr. Gaurav Kumar Gangopadhyay enlightened us about the various applications of plant tissue culture and plant biotechnology. Therefore, I propose sincere gratitude from the organizing committee to all of them for such interesting and thought-provoking lecture, making this session interesting and meaningful. I would like to express our profound gratitude to our respected principal, Madam Dr. Moshumi Singh Sengupta, for her valuable presence in this online lecture series, along with her constant support and suggestion in every minute matters that helped us to make this session reach such great heights of success. I would like to mention a special thanks to the honorable members of Sri S.S. Jain Sabha and all of the members of governing body of our college, without whose huge support it would not have been possible for us to organize this online lecture series. I would also like to thank our beloved IQAC coordinator, Dr. Shuchishmita Das, our Shuchishmita Di, for her moral support and guidance. I am very happy to express my heartfelt gratitude to all the faculties of our Department of Botany, Dr. Saurabh Ghosh, Dr. Anindita Singh Roy, Dr. Orpita Mukherjee, and our lab assistant, Mr. Kaushik Mondol, for their constant support and tireless job to make this online lecture series such success. I would also like to thank all our colleagues, especially Mr. Shomotonu Odhikari, Head of the Department of BBA, and Mrs. Rituparna Kundu, Head of the Department of Computer Science, for their constant technical support. Last but not the least, I would like to thank all the participants without whose presence these online lecture series would not have been meaningful and successful. So once again, I thank you all for your cordial cooperation. Thank you.
Thank you. Thank you, Rimi, Saurabh.
Thank you,
Thank
ma'am.
you, participants. Okay.
Thank you, ma'am. So
we
with
conclude the
this,
session, yeah?
yes, we'll just conclude the session. There are some announcements. After that, I'll just conclude the session. So thank you, ma'am. With this, we all come to the end of the session and the lecture series. It was great to have all of you supporting us, enjoying the lectures and participating so actively. The links for feedback have been provided in the chat box of Google Meet and YouTube Live platform. I'll just add a reminder that the form will be used for generation of e-certificates. So be very careful about filling up, especially the name with the salutation, the spelling of the name and everything, and the name of the institution. The form will remain active for 24 hours from now. Looking forward for your such active participation in our future programs and events, we call this session closed. So let's finally wrap up. I, with the permission of the convener, Dr. Rimi Datta, and the chairperson and principal of our college, Dr. Moshumi Singh Sengupta, would like to declare the session and the program closed. Ma'am, if you please close the session for us.
Yes, I close the session.
Thank you,
I
ma'am.
declare the official closure of this session. Okay. Thank you once again to all of you.
Thank you.